So, how many of you guys have heard about familial dysautonomia? Yeah, very few of you. That's because it is a rare disease. Only around 350 people in the entire world have familial dysautonomia. There's a 50% chance of surviving past the age of 40. There's currently no cure for familial dysautonomia, but they are working on treatments. 1 in 27 Ashkenazi Jews carry the gene. But since it is recessive, that doesn't mean that if you're a carrier that you have it. But just because it is rare does not mean it is not significant. In fact, my cousin has familial dysautonomia. It takes a great toll on her and all of the family around her. And we've had to watch her grow up and overcome the challenges and complications of the disease. So what is familial dysautonomia? It is a recessive rare genetic disorder that is a dysfunction of the autonomic nervous system. And the autonomic nervous system is the unconscious bodily functions that we can't control, but that our body does anyway. Some symptoms are the lack of production of tears, trouble breathing and digestion, problems regulating blood pressure and regulating body temperature, and a decreased sense of pain. So my paper is called Phosphatidylserine Ameliorates Neurodegenerative Symptoms and Enhances Axonal Transport in a Mouse Model of Familial Dysautonomia. Or, in simpler terms, drug lessens the symptoms of familial dysautonomia in mice by enhancing neural activity. So before I dive into the paper, I just want to get some vocab out there that'll come up throughout the presentation. The first is alpha-tubulin, which is a protein involved in forming the microtubules in cells, which then forms the cytoskeletons of cells, which provides the structure of the neurons. ICAP is a protein that promotes alpha-tubulin, and it is one of the proteins that is most downregulated in familial dysautonomia. HDAC6 is an inhibitor of alpha-tubulin, and it is upregulated in familial dysautonomia. The DRG is the dorsal root ganglion, or a cluster of sensory neurons in the spine. And throughout the paper, I'm going to be looking at experiments that were done in the forebrain and the dorsal root ganglion. Exon 20 is a specific piece of genetic code that's needed to code for the ICAP protein. And in familial dysautonomia patients, they're missing this exon. And to simulate a familial dysautonomia patient, they created the CKL knockout mouse model, where exon 20 is taken out of the nervous system. So just the presentation, ICAP is good, you want that. Alpha-tubulin is good, and HDAC6 is bad. So first the scientists had to create the knockout mouse model for familial dysautonomia. And they bred two types of mice, one here, and one with the Cre gene. And through the breeding, half of them will turn out to be familial dysautonomia knockout mice because the Cre gene is activated, which results in the deletion of exon 20. And when there's no exon 20, you have a familial dysautonomia mouse. And the other half will be a wild type control mouse. So for the CKO mouse mice throughout the paper, that is these. And before being able to test any drugs or anything, the scientists needed to prove that the mice were similar enough to human FD patients to be an accurate model. And they started out by comparing it, or taking observations of the weight of the body and the weight of the brain. And all these experiments were done in a lab in Tel Aviv, where they have a lot of information about FD patients, human FD patients, and they already know all these data figures on human FD patients, so they know what to compare to. In a human FD patient, the weight of the body and the weight of the brain is decreased, and that's what comes up in the knockout mouse here, and it's proven to be correlating to that. Next, they looked at brain protein expression levels, and these are all proteins that are known to be downregulated in human FD patients' brains. And they all show that the ICAP levels are decreased, which is the protein that is affected by familial dysautonomia. And all of these are downregulated in the mouse model, so it shows that it's an accurate model. The one exception is the NGF, or the nerve growth factor, but they said it wasn't enough to make this model not accurate. This is the same thing, but in the DRG, because they're both very important places to see all the protein expression levels. And same thing, the protein expression levels are same as the human, so it's an accurate mouse model. They next looked at the DRG protein development, and up here, this is the control mouse proteins in the DRG, very developed, and down here is the knockout mouse. And as you can see, there's some proteins that are a lot less developed, especially the ICAP, because it is the most targeted in familial dysautonomia. And these are representations of the figures showing that the cell count and the total DRG size is much less developed in the knockout mouse than in the control mouse. They then took a look at the forebrain neural branches, and in an ideal human, you want the neurites to be short and very strong signals, so that it's an intense signal that gets there the fastest, and it has the shortest distance to travel. But in FD patients, the branches are longer, there are more branches outwards, and the signal is weaker, so that the signal is also delayed. And here they noticed that the intensity in the knockout mouse was also decreased, and the length of the branches were increased, so it's accurate. They then took a look at the same thing, but in the DRG, and they also took into account the number of branches per cell. So if the branches are branching outward more, it's not as direct of a pathway for neural signals. And this all correlates to the speed of the neural signals, and the knockout mouse speed, or the neural signals in the knockout mouse's speed, is half of the control mouse's speed, and that's because the branches are longer, more branches outwards. Weaker signal takes longer for the signal to get there. They then looked at the percentage of alpha tubulin that was acetylated in the DRG and the forebrain, and in the human FD patients, the amount of acetylated alpha tubulin is decreased, which provides for the microtubules to be decreased, and therefore the structure of the cytoskeleton of the neurons are decreased, and the cells are more likely to collapse. And these graphs are showing that the knockout mouse also proves this and shows this. So up to this point, the scientists have just proven that this mouse model is very similar to the symptoms and traits of a human FD patients, and that they can do testing on it, further testing to test the drug. So this is the drug, phosphatidylserine, and this is where it's introduced into the mice. So the first thing they tested was the levels of ICAP protein and HDAC6 protein after introducing the drug. And 24 hours after introducing phosphatidylserine, they noticed that the ICAP protein levels were increased. And since ICAP is a promoter of alpha tubulin, that means the acetylation of alpha tubulin was also significantly increased. And then they looked at how it affected the HDAC6, and that was decreased. And since that's an inhibitor of alpha tubulin, the alpha tubulin was also increased here. So this is proving that the phosphatidylserine is both a promoter of alpha tubulin, ICAP, and an inhibitor of the inhibitor of alpha tubulin, HDAC6. They then looked at the neural branches with the drug, and the light purple is showing the knockout mouse without the drug, and the dark purple is showing the knockout mouse with the drug. So with the drug, the neural branches are shorter and there are less branches outwards, making for a more direct signal for the neural signals. And this affects the speed of the neuron signals, because the average speed of the knockout mouse with the drug's neural signals were 25% greater than that of the knockout mouse without the drug. So the drug is doing what it's supposed to do. It's increased alpha tubulin levels. It's made the neural branches shorter and less branches, and it's gotten the neural signal there faster. And this is the final diagram of what the drug itself and how it interacts with the rest of components. So phosphatidylserine activates or promotes more expression of the ICAP protein, and since ICAP is a protein that promotes the acetylation of alpha-tubulin, phosphatidylserine promotes the acetylation of alpha-tubulin. But it is also a regulator of the HDAC6 protein, which is an inhibitor of the acetylation of alpha-tubulin. So not only does it promote the promoter, it also regulates the inhibitor, making for the most acetylated alpha-tubulin possible. And with more alpha-tubulin, the microtubules are built up more, and the cytoskeleton or structure of the neurons are stronger, making for it to not collapse. So what's next in this particular study? So first they need to do further testing to see if phosphatidylserine works on human FD patients and not just the mouse model. And they need to do this by investigating these pathways more, and maybe they can even see if this drug is applicable to other more common disorders. So before I go on to explain why studying a rare disease like this is so important, so I want to venture a guess.
Well, like, perhaps you could find out information about like the creation of cytoskeletons, or like just figure out more about the cell itself.
Yeah, that would be, that's a good example of what you can do with this study. Yeah, exactly. So why is it important? So besides the significant toll on the people affected, of course, it can help scientists learn about pathways of more common disorders. And like Alessia said, in this specific situation, you can learn about the cytoskeleton and stuff like that. You can also, familial dysautonomia has neurological symptoms that are very similar to symptoms in anxiety and depression. And by studying familial dysautonomia, they can learn more about the symptoms and the treatments that they could possibly make for it that could possibly be used for anxiety and depression later. So there was an article about why rare diseases are so important in general, and they brought up two main points, or main rare diseases that led to discoveries in more common disorders. The first one they used was congenital leptin deficiency, which is a rare disorder where the leptin is not produced properly in the body, and leptin is a regulator of fat in the body. So through this study, they discovered vital information pertaining to the obesity crisis going on today. And obesity affects over 2 billion people in the world. So by studying just this very, very rare genetic disorder, they found out information that could help billions of people. The next one they talked about was familial hypercholesterolemia, which is a rare disorder where patients have dangerously high levels of cholesterol. And through this study, they produced statins, which are very commonly used today and prescribed today to lower people's cholesterols. And just in general, through the study of genetic disorders, scientists have learned a lot about the pathways and how genetics just work in general.
Do you know what else HDAC6 regulates?
Not outside of this model, but one thing that was brought up that I didn't really explain, but that we brought up was that HDAC6 is not just like a direct inhibitor of acetylation. and then this doesn't really show it that well, but it just regulates the acetylation of the alpha tubulin, and but in FD it's like upregulated a lot. So this is also not a direct inhibition. It regulates the amount because if it's inhibited completely, there'll be like too much cytoskeleton, and you don't want that either.
Next week we will be having our very own mentor present. This is Jared Boat, so come out next week to see what she's going to be presenting on.
Yeah, Jared.